Good evening, everyone. My name is Nilanjan Mukherjee, and today I will be presenting our research on the identification of novel immune synapse localized proteome for immuno-oncology using microscope-induced targeted photobiotinylation. Now, here in this study, we implemented our SynCell microscope, which is an innovative system that integrates microscopy, machine learning, and targeted photobiotinylation to enable precise and spatially specific enrichment of immune synapse proteins. So the workflow initially needs sample prep and AI-guided pattern generation, followed by identification of specific ROIs, followed by which a two-photon laser guides the precision photo labeling, and then we perform multiple rounds of photo labeling, which is automated, which leads to final protein enrichment and digestion through a streptavidin biotin pull down, followed by mass spectrometric analysis. And this is how our system or how our platform looks like. We have an electronic box, what we call it. We have the two photon laser right here, and then we have our optical box, which has dichroic mirrors, which allows this laser to pinpoint on the on the stage where our samples sit. And this is a epifluorescence microscope. Uh, and then we have our computer, which has the Autoscoop software installed, which allows us to perform the photo labeling as well as certain image and processing algorithms. So here we demonstrate the ability to perform photobiotinylation in subcellular compartments with high spatial accuracy. So in panel A, you can see both top-down or side views, or what we call it as the X, Y, and Z views of photo-labeled subcellular compartments, stained with their respective marker, like DRAC5 for nucleus, NPC for nuclear pore complex, NCL for nucleoli, and G3BP1 for stress granules. And you see that how nicely the photo labeling precisely falls on the DRAC5 signal. And you can see that from the merged images on each of these subcellular compartments. Now in panel B, uh, it shows confocal images of stress granules under photo-labeled and unlabeled conditions. And these images help us assess how photo labeling is specifically targeting stress granules in response to user-defined conditions. And the contrast between the photo labeled and the unlabeled samples gives us an indication of how efficient the photo labeling is and how specific the photo labeling is. And in panel C, you can pretty much see another set of confocal micrographs, but this time it highlights the precision and accuracy of photo labeling within the stress granule. And the images confirm that the photo labeling is both highly localized and reproducible, providing confidence in the technique. And to end with the volcano plot onto your bottom right, bottom left, you can clearly see that in the volcano plot, it shows the relative protein levels in both photo-labeled versus uncontrolled. And the proteins that are significantly enriched in the photo-labeled samples are shown in green. And these results confirm that the photo labeling got successfully performed, and this will lead us to isolate the proteins of interest within the stress granule. Now let's explore how photolabeling can be applied to study the immunological synapse, which is a very critical interface for T cell activation and communication. Now, on the left, you can see a T cell synaptic contact. And this is a well-established system. It has been used to study the formation of different immunological synapse, where T cells engage with antigen-presenting cells to coordinate immune responses. We move to the center. Here in the center, we perform machine learning, which allowed us to recognize and highlight the immune synapse region in white within a T cell spreading array. So you can see that how particular stain allowed us to create the photo labeling region. And this automated approach ensures precise detection of the synapse, reducing any kind of user bias. Then on the right panel, which shows photo labeling biotin in green, so as you can see in green, and it specifically marks the immune synapse protein. So you can pretty much see the top view, the XY view, and this is the side view of the different of the specific region, the immune synapse region. And the side view confirms that the labeling region is co-localized with CD3, which is a well-known immune synapse markers, and which reinforces the accuracy of this labeling method in capturing key synaptic proteins at the bottom of the cells. And here on the panel B, you see the T B cell conjugation model, and which shows us how the T cells interact with the B cells to facilitate antigen presentation and generate an adaptive immune response. On the left, you see the schematic diagram that you can see here, that the T and B cell synapse, and in the center, again, we perform the machine learning, which allows us to identify the immune synapse region shown in white. So this is a very small, nice region here. As you can see, this is the immune synapse region. Um, and finally, on the right, we see photo labeling in green, um, marking the immune synapse regions within the actin ring. So you can pretty much see that the green is right here. That's the biotin, and the photo labeling is done exactly where the machine learning generated uh, the photo labeling mask. And this confirms that the labeling is both highly specific and localized, further validating the robustness of this approach in studying the immune synapse dynamics at the molecular level. Now, we did a bunch of analysis, the protein abundance distribution analysis in the immune synapse proteome study. So as you can see on the panel C, we see the distribution of overall protein abundance measured as the ratio of photo labeled to control samples, which clearly showed or clearly gave us a broad overview of how many proteins are enriched in the immune synapse proteome. And specifically, we highlight key receptor proteins like the TCR and the BCR, and we also have the immune synapse protein, which is marked in green. And as you can see, that how statistically significant these immune synapse proteins are, and how their relative abundance is high in photo labeled samples versus the control samples. Now, as you move from panels C to D, um, we saw that in that T cell B cell interface, in the T cell B cell conjugation interface, um, certain proteins were successfully identified, right? And how these proteins can be mapped to the T cell region, to the B cell region, and this is how we kind of like how these proteins are being clubbed together coming from the B cell, coming from the T cell. And finally, on the panel E, we have we categorize these proteins, and then we identify like which particular synaptic cluster are these proteins coming from. So as you can see, that certain proteins certain proteins from that immune synapse proteome represents PSMAC, which is the peripheral SMAC. Some proteins represent the C-SMAC, and some represents D-SMAC. And then there are some other proteins, in addition to these, well-characterized immune synapse clusters like PSMAC, C-SMAC, and D-SMAC. We also identified a broader set of immune responses, a broader set of immune synaptic proteins, which are classified as the other immune synapse proteins. And finally, we did some gene ontology analysis of our enriched immune synapse proteins. So in panel F, we see results from our gene ontology analysis, which are applied to 69 enriched immunological proteins, which allowed us to categorize these proteins based on their cellular compartments, biological processes, and molecular functions. Our panel G identifies the list of top 20 non-immune synapse proteins that may have previously unrecognized roles in immune synapse dynamics. And our platform suggests that these may be functionally relevant in T and B cell interactions. And finally, on panel H, you see some protein-protein interaction network analysis we performed using String, which is a widely used tool for visualizing protein-protein interaction networks. So we took the 20 putative immune synapse proteins along with the 69 enriched immunological proteins, and we performed the string analysis to help us understand what particular network do these belong to. So we clearly saw that these interactions provide insight into how these immune receptors or signaling molecules or cytoskeletal proteins work together to coordinate certain immune responses.